At Collaborations Pharmaceuticals, we use real drug discovery intelligence alongside artificial intelligence to develop clinical candidates for rare, neglected, and unmet therapeutic needs. Our core expertise extends to high-quality data curation and machine learning model building, which we offer as a service. We can apply our technologies to drug discovery and ADME predictions. We can also conduct in vitro testing in our own labs and coordinate in vivo testing through our collaborator network. We have utilized drug repurposing to get us quickly to promising molecules, as we have demonstrated for Ebola, Chagas, and Alzheimer's disease. And we can apply these approaches to virtually any disease. We are unique in that we publish our own results in peer-reviewed journals and share our data models, unlike other artificial intelligence companies. Please get in touch to learn more about how we can help you at collaborationspharma.com.
Hello, I'm Sean Eakins, CEO and founder of Collaborations Pharmaceuticals. Thank you all for joining us today to learn about our MegaTox software and how we at Collaborations Pharmaceuticals can help you with your projects. I'll be showing you how we are committed to using computational approaches for toxicology, pharmaceutical, agrochemical, and consumer product uses. I also realize the value of your time, so I'll ensure that the presentation keeps to about 25 to 30 minutes and no more. So I have just a few logistical points before we begin, or I begin. If you would like to ask questions, please use the icon at the top of the screen, shown with the logos here. You can click on those at any time and write a question. We'll also have time at the end as well for you to write something down. In addition, you can also write your question in the attendee chat on the left of the screen. Thank you all for attending the webinar. If you are unable to follow or you find the quality isn't all that good during it, we will be recording it so that you can look at it again later. So if you miss something, you'll be able to look at it later on. So just a little bit of a brief outline before I get into the presentation. I'm going to give you an introduction to my company, Collaborations Pharmaceuticals, and what we do, and perhaps also address some of the challenges that you all may have. I will outline then the machine learning approaches that we take, and then describe MegaTox, our software, and provide a few examples. Finally, I'll discuss some of the future development areas that we're working on, and then I'll finish off by talking about Collaborations Pharmaceuticals and how we could help you with some of our client use cases as examples of what we've been able to do. Finally, we'll have a question and answer session, so get thinking about those questions now. For those of you that don't know me or have not read any of my papers in the past, my name's Sean Eakins. I'm the CEO of the company. I'm an expert in applying machine learning to various drug discovery and consumer product applications. I have over 24 years of research and development expertise in the pharmaceutical industry, and I've been able to publish over 300 publications and numerous patents with various collaborators. I've also been fortunate to edit and write a number of books and articles around computational toxicology, and one of those is shown here. In addition to these roles, I've been able to consult for many other companies as well in the pharmaceutical as well as consumer product space. 5 years ago, I founded Collaborations Pharmaceuticals, and we're located on North Carolina State University campus. Currently, we don't have any angel or VC funding, and so we're pretty flexible in the types of projects that we work on. But we have been able to obtain over $7.6 million of NIH and Department of Defense grant funding from the US government. This has allowed us to develop our machine learning technologies and software platforms, as well as push forward our drug discovery projects. To date, this has resulted in 9 orphan drug designations from the FDA for both rare and neglected diseases, as well as 3 pediatric rare disease designations. So enough about me and our company. What are your challenges? I know in the consumer product space, if you're working on cleaning products, perhaps some of the areas of interest based on projects we've worked on previously, may be in prioritizing your ingredients for their potential to disrupt the endocrine system. If you're working on cosmetics, clearly you're all interested in non-animal testing options, looking at skin sensitization models, etc. Others may be interested mostly in the environmental impact of their compounds. So can we look at the biodegradation or bioaccumulation? In the pharmaceutical industry, we obviously have many toxicity endpoints that we're really concerned about, such as cardiac or liver. And I'll be talking about some of these examples today. And my long history and background in drug-drug interactions is obviously still a major concern, especially in the older populations that are taking many drugs and therefore increasing the probability of drug-drug interactions. In the agrochemical space, your molecules and their biodegradation will be important, as well as toxicity to non-target species. Finally, in the animal health realm, obviously you're challenged by being able to develop treatments mostly cost-effectively, as well as all the above concerns, such as toxicity that we'd see with pharmaceuticals, as well as their impact on the environment. A nice review of some of the opportunities of computational toxicology in these various areas was published last year by the EPA, and I referenced that on this slide, so I point you to that. What we've been able to do at Collaborations Pharmaceuticals is develop a number of technologies and model collections. One of these is our Assay Central software, which is our core technology for both the curation of data and building and validation of models. Today, I'm going to talk about MegaTox, which is really our subset of toxicology models and molecular property models. In future webinars, I will be presenting on MegaTrans, which is a subset of our human drug transporter models, as well as MegaPredict, which is our collection of thousands of models for both targets and diseases that have been assembled from the Chembl database. In terms of building machine learning models, I will not dwell on this into too much detail. I would refer you to some of our earlier webinars that we've put out this year and that are available on our website. But briefly, we've focused initially on using the Bayesian algorithm along with the ECFP6 fingerprint descriptors. We perform ordinarily 5-fold cross-validation, and we've been able to use this approach for thousands of data sets. In addition, we've applied many other standard machine learning algorithms, and we've been able to do comparisons, which I'll show you in a few slides. We're able to wrap up various models and share them with our collaborators using our software, and we have useful visualization technologies so that we can look at the role of particular features on a molecule in association with a particular endpoint. And so the atom contributions can be highlighted, as is shown on this figure. And we can also build not only classification models, but also regression models with some of our latest algorithms. Before I get on to MegaTox, I'm going to highlight a few particular tox applications of various models that we've built. If you are interested in the underlying machine learning technologies, I would refer you to some of the papers that we published as long ago as 2015 on using the Bayesian approaches. And many of the examples I'm going to show today have already been published this year. These include the acute oral toxicity in rat models, as well as the drug-induced liver injury potential. I'm also going to describe some of our cytochrome P450, as well as hERG inhibition and blood-brain barrier work that hasn't been published yet. We actually have hundreds of toxicity models that we've built and that can be wrapped up and provided as a custom selection of models. These obviously include some of the larger public data sets that we've built models for, including the EPA data sets, Tox21, ToxCast, etc., as well as models that we've curated ourselves or we've taken from other published data sets. Most recently, we've published a large-scale comparison of over 5,000 Chembl datasets, and each of those datasets has over 100 molecules in each. And we've performed comparisons with different algorithms, including support vector classification, decision trees, various Bayesian methods, as well as deep learning, k-nearest neighbors, and random forests. We've assessed 5-fold cross-validation statistics, as well as performed external testing on various datasets. This quite rigorous approach essentially has been published most recently in Molecular Pharmaceutics, where we describe it in more detail. But in summary, what we found was the Bayesian approach that's used in MegaTox performs as well, if not better, than many of the other methods. And we're using here a rank normalized score to visualize all of the model metrics for the 5,000 models in each model case. And we've also used a rank normalized top score, so a difference from the top. And you can see that the Bayesian approach is on a par with support vector machines and deep learning, as well as some of the other methods. So the bottom line from this comparison, and why we've focused primarily on the Bayesian approach, is because obviously the computational complexity of some of the other approaches really doesn't give us much of an edge when we look across these thousands of data sets. So we may get better value for our money in terms of building Bayesian models versus some of these other tools. We've also been able to perform a number of external tests, in particular focusing on toxicology endpoints such as the pregnane X receptor, PXR, as well as the ion channel hERG. And these are examples of external testing, prospective external testing that was done with molecules that we have been able to test ourselves, and also comparing the predictions that are generated with both manual curation models as well as auto curation. And what we basically found in these two examples is that they're comparable with each other in terms of the predictive nature. And obviously the benefits of using an auto curation type of approach are in terms of the speed with the data sets. So some of the other models that I mentioned earlier that we've focused on this year include drug-induced liver injury, or DILI. This is obviously a major problem for some companies, especially as most of the unpredictable adverse reactions that occur to drugs are due to this. And this is also a leading cause of post-marketing drug failure. So there's a high cost if your molecule is pulled from the market because of this. So ideally, it would be fantastic if you could predict this as early as possible before you get into the clinic. And there have been a number of computational models to predict this property. In 2010, I published perhaps the first computational model on drug-induced liver injury with a small data set of several hundred molecules from Pfizer. And there have been many examples and publications from the FDA, including those of Weida Tong and many other groups in the intervening years. And most recently, both AstraZeneca and Pfizer have published a couple of papers where they had made a number of data sets available for 100 to 200 molecules. And obviously, in the intervening decade, there's now over 1,000 molecules that are being curated by the FDA that have DILI data or don't have DILI data in terms of activity. And we've been able to use these data sets to produce an array of models, and this work was recently published in Molecular Pharmaceutics. And just as an example, one of these models built with the DILI rank data had excellent five-fold cross-validation values, ROC of 0.8, sensitivity of 0.74, specificity of 0.75, and accuracy of 0.74. And these models perform very well on external testing as well, as we were able to predict a number of drugs that were approved over the last few years, and several of those obviously had been found to demonstrate DILI. So what we actually have now are capabilities where we can use just the structure of a molecule to predict this quite complex toxicity endpoint. And I believe we've progressed significantly further than where we were about a decade ago, both in terms of the amount of data that's in the public domain for building these models, as well as in the quality of the algorithms that we can produce. We've also been involved in a consortia over the last few years that's been organized by the NCP, Interagency Center for the Evaluation of Alternative Toxicological Methods, and the EPA's National Center for Computational Toxicology, as it was called then. This was involved in using a few data sets around rad acute toxicity, where the LD50 data is known, and there's categorical information around the various data sets of thousands of molecules. We were able to build 8 models in total with different thresholds and compare different machine learning methods that are shown on this particular slide. I know that the consortia paper has now been submitted But we also have submitted our more recent evaluations of this data set, both the training and testing, and also evaluated accuracy of other external data sets that have been related to rad acute toxicity. And this work was published very recently in ACS Sustainable Chemistry, so I'd refer you to this. What I can say in summary is that, again, the Bayesian approach is comparable to deep learning and KNN when we look at these 8 different model data sets. A final data set that I'm going to show you here relating to blood-brain barrier penetration used compounds from a previous paper with over 2,000 molecules. And as you can see with the 5-fold cross-validation, we have a fantastic statistics, 0.94 on the receiver operator curve. And then other model statistics were very high, generally greater than 0.7. We've used this model with an external set of compounds that are not in the training set. And these molecules were published in 2010 by Pfizer and used as the basis of the multiple parameter optimization to predict blood-brain barrier penetration using log D, log P, pKa, molecular weight, total polar surface area, and hydrogen bond donor counts alone. And what we found was that our machine learning model, which is a Bayesian model, was able to predict 37 out of 40 of these BBB active molecules. That represents 92.5% of molecules. In comparison, the MPO score, which has been widely accepted as a measure of blood-brain barrier penetration, is only able to correctly predict 72.5% of these molecules. We're currently evaluating these models with larger data sets, and we're working with a partner organization that has expertise in blood-brain barrier penetration. And so I would imagine that next year we'll be able to publish a new manuscript on this. And our goal is really to validate the machine learning approach versus these more rule-based type methodologies. Let's move on to the ecotoxicology situation. Obviously, there are many data sets that are starting to be put onto websites, such as the EPA's, and we've taken one of those here for bioaccumulation. This has about 670 or so molecules. You can see that the training set data on the left-hand side has an ROC of 0.92, and quite useful in this regard, they provided a test set that could be used, and we've been able to use this with our model. And you can see again, we get very comparable ROC values of 0.94, as well as very similar statistics to the training set, indicating that this is unlikely to be overtrained. So this is just one of many ecotoxicology data sets that we can model. And I think it's a nice example to demonstrate that the technologies that we use in the ECFP6 fingerprints and the Bayesian tools can be applicable to all different kinds of data sets, not just human toxicology or animal toxicology data. Because of my 20-odd years of experience in drug metabolism and drug-drug interactions, I'm very interested in seeing how our machine learning approaches can model P450 metabolism. And there are numerous data sets out there that are in the public domain, and we've been trying to make use of them and recurating them to clean them up. And this slide just demonstrates some of our earlier work in this field, collecting the data sets for the individual P450s, and then using them to do comparisons with the different machine learning approaches. And as we've seen with all of the different validations we've published so far, again, the Bayesian approach is, if not ahead, it's comparable to other machine learning approaches when we look at the different statistics that we ordinarily look at. Again, suggesting that these approaches and building classification models may be very useful. We can also look at in vitro data, such as human liver microsomal stability, and now there are plentiful data sets in databases such as Chembl. In this example, we have over 3,000 molecules, and you can see the model statistics are about 0.84, and all of the other statistics are greater than 0.7, apart from the MCC and the kappa, which are about 0.5. So these are very good model statistics for building classification. And we are using these actively with a lot of our drug discovery projects, both internally as well as externally with collaborators. We also are able to model microsomal stability in other species, and we've been curating those data sets, and we've recently published some of this work as well with our collaborators. As you can see from these examples, which are really just some of the highlights, because of time, I'd obviously love to go into the other hundreds of examples, but we're certainly focused on the future. We're currently implementing additional machine learning algorithms, and this is something that we'll obviously be publishing in due course in terms of comparisons with the different data sets. And we're also looking at the future, far future. And one of these areas is obviously quantum machine learning. And over the past year, we've been taking a look at that. So I have a slide to show you some of our work on that. We're also interested in optimizing compounds to remove toxic liabilities, and this would obviously be very interesting to some of your companies. And we're using multiple optimization generative models, and I have a slide to show you that as well. So here we go. In terms of quantum machine learning, we're very interested in using this approach, and we have, with collaborators here at NC State, been evaluating the IBM QHub 53-qubit machine in terms of how this performs with large data sets. And obviously with very compute-intensive approaches like deep learning, these require obviously a lot of time to generate the models. And you can show here on this purple line, we're showing with increasing dataset size how it scales in terms of time to build a model. And it's pretty linear. And so if you imagine that as the data sets get bigger into the hundreds of thousands or millions of molecules, we're going to be waiting a very long time just to build one model. So now imagine if you have 5,000 or so of those models to build. It's obviously never going to happen, right? So we've been looking at the quantum machine learning approach, and this is a simulation with real data sets to show how quantum computer can actually pretty much max out in terms of the amount of time to build models. So it's no longer linear. We published this work in a preprint, which is out at ChemArchivics, so you can take a look at that. And we're in the process of looking at the reviews of this as it's actually out for publication at the moment. We've done a number of other evaluations of classical computing versus quantum computing, and so I would definitely refer to this if you'd like to find out more about what we've been doing. The next area of interest, as I mentioned, is how can we remove toxicity and come up with new designs for molecules. So for example, if we had a molecule that inhibits a target of interest, but it has a liability, such as CYP inhibition, how could we now come up with analogs that maintain the activity of the target, but remove the toxicity? And there's been a lot of interest recently, in the past couple of years at least, in recurrent neural networks and generative approaches. We've also been developing tools that are more rule-based for analog generation. And so one of our ideas is how can we bring these two approaches together? So our approach here is showing the recurrent neural network approach, where we have an input molecule, we have the parameters, we have the data sets, the machine learning models that we've built, both the target and the activities, and we use these to come up with molecules and limit them based on the model scores. And we've also developed synthesizability scores based on literature information. And internally, we have the capabilities to do retrosynthetic analysis. So once the recurrent neural network comes up with the molecule designs, we can immediately then do the retrosynthetic analysis and figure out, well, actually, this is how we would make the molecule. So this may be one way that we can optimize molecules. And clearly, this is just one example in the toxicology domain space, but you could definitely think of many other applications. So how are we differentiated from others, especially those companies in the AI space? Well, as I mentioned at the outset, we don't have VC funding, so we have complete freedom to operate. We can go basically where you want to go in terms of projects. So therefore, your intellectual property remains your IP. We have no interest in having any access to that or any reach through. So our real concern is just delivering the quality results that you need, applying our technologies and focus, which is primarily where we separate ourselves, I think, from the competition, is on our data curation and external model validation approaches. These are at the very heart of what we're doing with the company. In addition, I think an important differentiator is the experience that we have in drug discovery and computational ADME tox, and that perhaps sets us apart from other companies which are more focused on the algorithms and tweaking those just to try to get a little bit of an edge. I think we're a bit more pragmatic and we want to put these things into action. The services that we can offer alongside obviously what I've shown you today include the dataset curation, machine learning. We obviously have thousands of models that are ready to go, and we can apply those in drug discovery and disease areas, as well as ADME and tox. We can perform predictions and cheminformatics analysis of your libraries or ingredients, and it doesn't matter if the training sets for the models that you have are small, on the order of less than 50 molecules, or large, on the order of hundreds of thousands. I think we'd be able to cope very well with them. And we have a lot of experience with predicting very large libraries in the order of millions of molecules. We can do things like prioritize compounds for synthesis or prioritize them for in vitro or in vivo testing. Those would be a few of the uses that we've already been experienced at doing. We can also come up with ranked predictions with chemical diversity constraints and take into account many different models at the same time using Pareto optimization. We also have access to models for molecular property calculations. And as I mentioned, we've got our own software now for synthetic feasibility and retrosynthetic analysis. So we're very happy to take on any new challenges you may have. And finally, I just have a few real-world examples where we've been working with clients. One of these was a consumer product company that we've been working with for over 3 years now on endocrine disruption. We've looked at estrogen receptor, androgen receptor, and aromatase. To date, we've published 4 papers from this project. We've produced many posters and talks at major conferences. So this has been a very fruitful multi-project collaboration. We've also had a collaboration with another consumer product company also interested in endocrine disruption. And we have ongoing projects with a rare disease company now, looking at developing designs for new molecules to overcome an ADME issue. And this is a great collaborative project which has now gone through 3 cycles where we propose molecules and they've been synthesized and tested. And finally, we've been working with a small animal model CRO that's given us access to in vivo data, and we've been modeling various targets and ADME properties for them, as well as in vivo activity. And how do we work with companies? Well, this pretty much depends on the project scope and the number of people involved, whether that's curating public data or proprietary data that you have and you just want us to model it, or if you want us to design molecules and score them. For example, if you need us to enumerate molecules and figure out how to make them, we can do that. Most of the projects that we do, in fact, all of them so far, have been fee-for-service, where the client owns the IP entirely. We're obviously open to other ideas if there's perhaps a good collaboration involved. We've certainly done collaborations with academics where we're joint owners of IP. And depending on the extent of the project, we may have dedicated FTEs associated with your project. We're certainly open to longer-term multi-year projects as well upon negotiation, and our software is available to license with larger companies for your own in-house use. But primarily, we're not focused on developing the software for external use. I think our strong suit is really using this internally for your projects. I'd just like to thank the NIH and DoD for funding, and in particular the NIHS that's funded MegaTox. And I'd be happy to answer any questions. And so you can go to the little icon at the top of the screen, or you can add the questions in the attendee chat. If you want to contact me directly, you can send me an email at sean@collaborationspharma.com. And if you want to find out more about our company, you can go to www.collaborationspharma.com. My next webinar will be in January, and I'll be describing our pipeline. Okay, so I'm now going to take a look at your questions. All right, just like to give me a couple of seconds and I'll try to address them. We will definitely be making the recording available, and we can send that to you if you registered, obviously. And we won't be making the PowerPoint available, but obviously you can look at it in the recording. So one question around the naive Bayesian approach was about the univariate feature selection. And we have another question on HLM models from Kemble. Are there different assay differences from diverse sources? How are they overcome? That's a very good question. How do we take into account that data sets may be using different assay approaches? We can narrow down if there is detail in terms of the types of assays. We can certainly drill down into that kind of data and try to curate it. That is definitely something of importance when we're looking at trying to look at IC50 values, perhaps for a target versus KI versus other measures, percent inhibition. Obviously, we can narrow down to those particular areas. If there isn't any experimental data that's provided, we're just basically taking what's there. And I think, that's obviously a limitation. Some databases are much better curated than others. Kemble does a really good job of that. One question about solubility modeling. We haven't been involved in solubility modeling to date. We've tried to focus a lot on toxicology as well as drug discovery. But that's certainly something we could take a look at. Obviously, that would require us to focus more on regression models. And I would say so far, we've been putting a lot of effort into classification and especially Bayesian approaches. We have a question on the acute toxicity model. One of those integrated into the consensus CATMOS model. Yeah, we were involved in that particular consortia, and so our earlier models, I think, were incorporated into that. But subsequent to it, we've definitely added more since that particular collaboration. So I'd refer you to the paper because we go into much more detail than what we'd actually done for the initial consortia. And it's also a more updated model because the work we did for the CATMOS consortia was 2 years ago. All right, are there any other questions? If not, I'll try to hold on to what I said at the outset. I'll try to keep it to 30 minutes. I would like to thank you all for your questions and attending, and we can send you the link to the video if you'd like to watch it again. Again, just a reminder, you can contact me at sean@collaborationspharma.com. And the next webinar is in January. And if I don't hear from you from email, have a happy holidays wherever you are in the world, and Happy New Year to you all. Thank you.